Hey Pops, what's a CRISPR? Well...
Ever since the beginning of life on Earth, the microscopic war between bacteria and viruses persists to this day. Bacteria have rudimentary tools to defend themselves against viruses, but most are generally ineffective. However, in the event that a bacterium survives a bacteriophage attack, it is able to utilize a new form of defense, CRISPR. With CRISPR, the bacteria is more prepared next time the virus strikes. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. In bacterial DNA, there are the CRISPR repeats that are going to be short, equally spaced segments of DNA measuring 20 to 40 base pairs in length, and they're going to be palindromes, which causes them to form hairpin loops like tRNA. However, in genetics, palindromic DNA does not appear exactly like a regular palindrome. Instead, it is only considered palindromic if the complementary strand of DNA reads the same code if reversed. For example, the code CTATAG. Between the identical repeats, there are equally spaced bits called spacer DNA. Each segment of spacer DNA is unique in reflecting the bacteriophage DNA that serves as a record of past invaders. Now, when bacterial transcription comes near the CRISPR archive, first, a series of Cas genes produces CRISPR-associated Cas proteins, and secondly, the spacers of CRISPR are transcribed from the CRISPR locus as necessary, yielding pre-CRISPR RNA that is further spliced to form our final product, CRISPR RNA. When a bacteriophage injects its DNA, normally when the immune system fails, the foreign DNA hijacks the native DNA, spurring more phages and eventually killing the cell. But since the bacteria has a CRISPR system, it's going to create Cas proteins that form a Cas complex, and while further generating CRISPR RNA that is associated with the familiar bacteriophage DNA. The CRISPR RNA will fit into the complex, and the now greater complex will be used by the bacteria to fight that new viral DNA.
お前はもう死んでいる。何?
However, if CRISPR doesn't have a spacer that matches the new viral DNA, the Cas system needs to create a new protein that breaks it apart but more importantly saves a copy into the CRISPR system as a spacer between two repeats. One way scientists are manipulating CRISPR is by using the Cas9 protein. Cas9 is a nuclease that can cut DNA. To do this, CRISPR RNA, transcribed from spacers, fits in with tracer RNA that holds the CRISPR RNA in place, creating a complex where the protein can cleave and incapacitate the foreign DNA. Scientists connected tracer RNA and CRISPR RNA to get the tracer RNA CRISPR RNA chimera, more simply called guide RNA. With the guide RNA and Cas9 protein, CRISPR can deactivate a gene by breaking it and having the cell fix the DNA with the addition of mutations. If we wanted to insert a new gene, then the piece of the host RNA would be placed in Cas9 as it cleaves the bacteria's DNA and embeds the host DNA in the genome. The discovery of CRISPR was revolutionary because scientists discovered that it was programmable, in the sense that it could be modified to fight other viruses by modifying Cas9 with third-party CRISPR RNA. CRISPR is progressively becoming a new time-saving tool as scientists are answering age-old biological questions while bringing up therapeutic applications. The introduction of CRISPR has led to theories pondering the nature of its uses in the medical field. Scientists have already been able to cure rats of HIV, but human applications still lie on the horizon. For example, some believe that CRISPR can help treat cancer, make quick long-term changes to populations, lower the cost and time requirements of experiments, and most controversial, make modifications to human reproductive cells. CRISPR technology may change the way we view the world, as humanity is slowly but surely leaning to improve our lives from aging reversal to building superhumans.
Eat 'em up, beat 'em up, beat 'em up, beat 'em up. Eat 'em up, beat 'em up, beat 'em up, beat 'em up. Reese's Puffs, Reese's Puffs. Eat 'em up, beat 'em up, beat 'em up, beat 'em up. Reese's Puffs, Reese's Puffs. Eat 'em up, beat 'em up, beat 'em up, beat 'em up. I got Reese's Puffs in my bowl. Now my day's on cruise control. I got Reese's Puffs in my bowl. And just like that, I'm on a roll. Reese's Puffs, Reese's Puffs. Peanut butter chocolate flavor. Reese's Puffs, Reese's Puffs. In the AM, it's the flavor I savor. Peanut butter and chocolate too. You know how I do, that's what I wake up to. My Reese's Puffs inspired this rhyme. That peanut butter chocolate combination's on time. Reese's Puffs, 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 Reese's Puffs. Eat 'em up, beat 'em up, beat 'em up, beat 'em up. Eat 'em up, beat 'em up, beat 'em up, beat 'em up. I got Reese's Puffs in my bowl. Nowadays on cruise control. I got Reese's Puffs in my bowl. And just like that, I'm on the road. Reese's Puffs, Reese's Puffs. Peanut butter chocolate flavor. Reese's Puffs, Reese's Puffs. In the AM, it's the flavor I savor. Peanut butter and chocolate.